All right, am I audible and visible at this point?
You are audible, visible. We could see your slides. You're in good shape.
Fabulous. So I'm actually really ecstatic to hear what Adam and Varun have presented about this CDMS, the new CDMS product that it really is going to address some of the primary needs that we have as a research organization. And so you're getting very much our experience here launching LabKey as a new service here at Cincinnati Children's for researchers. And you'll see some of the journey that we've had here and how hopefully this new CDMS product is going to address our concerns. I've prepared this presentation based on our experience before knowing about CDMS. And so that's the perspective we're going to take here. But yeah, this is, let me see what is advance. Okay. All right. So very much a work in progress. We are less than a year licensed with LabKey, and we are still building our first primary project in LabKey and starting to roll out to some other research groups now within our institution. So what I want to accomplish today, I want to share what we're building. I'd really love to solicit any feedback or ideas, and if there's not enough time here, I have my my contact information at the end. I would love to get input via email or any other contact. And then also, you know, as you know, we're trying to build out features here. If there are things that sound like you guys could use at your institutions, it'd be helpful to get a number of users asking for the same things to deploy resources on the LabKey side of things. As Josh and Adam kind of mentioned about development priorities. Anyway, so our team here in the Division of Critical Care, we've got 6 PIs and at any one time 4 to 6 trainees with 7 research assistants and 1 biorepository manager. And then we've got a lab manager in name, but he is a jack of all trades that helps out with all sorts of aspects of our research and operations here. It's just amazing. So we focus all of our study on sepsis, septic shock, and what happens there with critically ill kids that come into our ICU. This is our primary research focus. So just to touch on this a little bit, sepsis is a potentially life-threatening condition where kids develop organ system failure in the setting of severe inflammation and infection. And sepsis is really a heterogeneous disease with kids that have mild varieties and others that get very sick and pass away. But up front, when kids present to the ICU, we don't know which bucket they're in, you know, and so we conduct research to do both prognostic and predictive enrichment to identify kids that have low risk of progression to organ failure and death, and those with high risk, the higher risk cohort meritorious of possibly higher risk therapies or more invasive interventions. And then also within that high risk mortality group, you know, we want to identify, you know, biological mechanisms that might respond differently to different therapies. And so we have a research program dedicated to identifying kids in different strata using various biomarkers and classification strategies. And so that's how we approach research. So when kids come into the ICU with sepsis or septic shock, they're approached for enrollment in our studies, and they, when consented, we start collecting clinical data. So all of our monitor data, vital sign data, laboratories, studies, all get collected. We draw blood samples from usually on day 1 and day 3 for a variety of different downstream analyses. From these biological samples, we'll run biomarker assays looking at different protein levels in the bloodstream. We'll do gene expression analysis. And from these analyses, we'll generate stratification scores. Prominent among these are the Persevere biomarker panel, which allows us to detect low, moderate, and high risk patients for mortality, and then sepsis endotypes based on gene expression in there. So So this is kind of the bread and butter of our research operation. And this program has actually been ongoing for 20 years. We have 20 years of patients' data and samples, along with associated analyses in our research group. So, like I said, we collect demographics, vital signs, all sorts of patient-centered data. We have information around samples that we need to maintain in terms of when they're drawn, how many aliquots, free soft cycles they have, and then all of our downstream analyses, which are now including, we're branching out into metabolomics, proteomics, methylomics, with the omics kind of progression technologies. So all sorts of information we've been trying to manage, and it's just become more and more difficult over the years to accommodate all this as the repository and clinical database has grown. The vision that we've had here is to integrate all this data. We want to pull it all together. We need to collaborate. There's a lot of internal researchers that collaborate within our institution, and then we have external collaborations with researchers around the nation. And so we needed to have a place that was easy for us to share data. And then ultimately, we want to generate knowledge from this information that can impact patient care at the bedside. And we publish that knowledge and share it with other investigators and physicians. And so that was the vision of all that. And my role with this group is I help out with the kind of the data management and creating data sets, and organizing all the information that we've had. Two of the requests that I most commonly get have been for me to generate a study data set from our repository. And these are really just numbers, and variables that have already been collected that people want to subsequently analyze. And so we'll include demographics, other clinical variables about their organ system failures, downstream analyses that have already been completed, including the biomarker panels, gene expressions, and then whatever stratification scores have already been generated. And so I'll pull that data together and create the data set for investigators to analyze. The other type of request that I often get is for sample polls. And I'll have an investigator say, hey, Steve, I'm looking at these two groups of patients characterized by these clinical features and these biomarkers and these stratification scores. I want all the available serum samples, or, give me the 400 sickest kids, with these parameters. I want to look at their gene expression, and I'm sorry, I want to look at their DNA, and so that I could look at genotyping or their RNA, for gene expression. And so I'll need to generate a pick list for these researchers. When I picked up this project, however, our data were scattered all over the place. We had some difficulties over the years with our old bioinformatics group here in the institution, helping us maintain our data in a unified location. And so we had clinical data that was scattered across multiple different Excel spreadsheets. A REDCap database had been stood up about 7 years ago that was capturing things adequately moving forward, but it wasn't really integrated with some of the old stuff, and it wasn't complete. We had all of our biorepository data spread between Excel spreadsheets. Some of the oldest stuff was still on paper in notebooks. And then all of our research analyses, again, scattered in different file folders on shared drives, raw data files living here and there. It was kind of all over the place. And so you had to talk with different people in the group to find the fragments that the researchers need to analyze. And I was charged with the responsibility of pulling all this together into kind of one unified location. So we looked at a number of options to figure out where we could do this. And as was mentioned with some previous presentations, there wasn't any one place that I found that was able to integrate the clinical, the biorepository, and the downstream research data, just by looking at a number of different options there. And until I encountered LabKey. And LabKey offered a platform where we could build some of these integrations and house our data that would allow for organization and analysis. And so we moved forward there with some testing, and then ultimately licensed LabKey earlier this year to start building this first trial project with sepsis genomics. So what did that look like for us on this side? Well, I had to find all the data files, and use some tools to sniff through our shared file structure to find all the fragments that I needed. Compiled the data. I'm most familiar with R in terms of a coding language, but able to clean, harmonize, merge data sets, identify discrepancies. We actually ended up having to rebuild our REDCap database to accommodate some guardrails and validation tools that we needed for the data that was being ingested from the clinical research coordinators. Then we actually went back through and manually logged all of our samples stored in the freezer so we could capture all of the barcodes, and identify a very granular location and data for each sample. I was familiar with with Access, and just threw that out on an Access file for different research assistants to log there. So we had all those different kind of parts that we were pulling together, and finally we were able to upload it into a new REDCap instance and LabKey. And I'm talking about both of these together because we wanted to continue to use REDCap as a point of entry for our clinical data. Our clinical study is a multi-site study. We have, I think, 13 plus different sites over the years that have contributed data. And everybody historically has become very familiar with the REDCap, forms that we've entered, and several of our researchers wanted to maintain that point of entry. So all the data, clinical data, comes in through REDCap, and LabKey has a really nice integration that sweeps in all the data from REDCap into to LabKey. The clinical sites will send us their biospecimens, and we're now logging those in the sample manager module. So again, clinical data comes into the study module on LabKey, and then the biospecimens are logged in sample manager. And that sample manager allows us to check out the biospecimens, do our analyses on them, and then we can check them back in and maintain provenance as to who's using them, where they're going, what are they being used for. And then we can upload all of the research data as analyses that are housed there. And we haven't built out any dashboards or visualizations yet, but we have been able to query across all these different things with much greater ease since starting to build out LabKey than we were ever able to do before. So what does this look like? We've got a bunch of different data collection instruments within REDCap, including an instrument that collects the samples, or that allows us to record the samples that are collected at the bedside, and that the CRCs pull there. This gets swept in. Okay, yeah. Here are two study folders in LabKey, but these get swept in block into the study module within LabKey there. And so we have our source of truth out in REDCap, and everything is replicated perfectly here. And we wrote some ETLs, and this is Patrick Lonnie, our lab manager, put these together to pull this every hour over from REDCap. Okay, but as Varun, I think, talked about as well, we wanted our sample manager to be able to, the biorepository manager, we wanted them to be able to link the samples that were being collected directly to a patient and a time point in our study right off the bat when those samples were created within LabKey. So Patrick wrote another ETL there to push information from study over to Sample Manager. I imagine this is going to be part of the integration that's been refined in the new CDMS platform that has been discussed here today. And so these ETLs are really just some XML that's telling LabKey what to push where. There's a little bit of a SQL that's written to pull out the variables that are necessary to identify the visits and the patients to create sources for samples in Sample Manager. Yeah, that gets pushed over here to Sample Manager, and we're able now, let me use this pointer here, to have our patients show up automatically here, and the collection days as a source type because of these ETLs that Patrick has written. And so our biorepository manager and research assistants can just come in here to the sources when the CRCs drop off new samples in the lab, and they're able to select, which patient and which day the samples are coming from, and they'll create new samples. And these come in as parent samples, and they can be any of the standard clinical tube types, gold tops, serum separators, EDTA, lavender tops, PAX genes for RNA. So we have all sorts of different tube types that are collected here. And then from these, once they're registered as parent samples, we derive child samples there, like RNA that's extracted from the PAX genes, or DNA from the EDTA tubes, their serum from the gold tops, and those are all logged, and we can identify very easily places to drop them into our freezers and specific boxes there, and these are linked back automatically to the study. And you can look back through the lineage and see, within, Sample Manager, you can find whatever sample you want in whatever box and see what the parent tube was, the day of collection, and the original patient. And this can be viewed again, from another view and see, all right, our patient is here at the top of the tree here. Then we can see the collection days and what other samples are available there. So this has really revolutionized our insight into what we have in our repository and what's available. And again, these are linked back up into the study module so that you can see what samples are available from within this side of the integration between the two modules. You'll see here as well that we have right now our downstream analyses that are just manually uploaded now. We're working on building out data sets for genotyping for gene expression, but we have biomarkers and some of our echocardiography and stratification stores already in there. In terms of how we're utilizing this currently and getting data out of it, okay, my vision is ultimately to have things set up so that individual PIs will be able to log in and build their own data sets without having to have technical knowledge about code or ETLs or any scripting. That they'll be able to come in here into a data set and use the customized grid function and then select the various different fields they want to pull together into their data set. Okay, and then use filtering and sorting to generate the specific cohorts they need, both to generate an analysis data set as well as to create a group that we can then select samples on, that they can go in and select samples on. But that currently isn't available right now. We're trying to find ways to pivot data. We have our biomarkers entered currently in a long format, but many of our PIs want them in wide so that it makes it easier to analyze for their studies. So we're working on SQL queries to pivot the data on LabKey and offer that option to them as a data grid there. But currently, we're relying on R to access the data, and I go in there and use R to pull the data out of LabKey directly. And LabKey has a really nice R package that allows me very quickly to sync or to link up to the data that's housed in LabKey using an API session key. And then I get direct access to the data tables. And in terms of writing this code, it's not very complicated. LabKey itself it generates an export script that you can then modify and refine, but it gets you started there within LabKey. You create the script and it spits out, hey, here's the command of the various functions, and you can adjust the arguments as needed here to achieve whatever you want in terms of the specific data you pull out. But so using R has been very helpful in terms of being able to access quickly the most up-to-date information while we're still building out our data grids for PIs to get in there and pull data directly. So what are we still working on? So I mentioned how we're trying to pivot data tables on LabKey long to wide. My ideal would be for an investigator to be able to log in and say, hey, from these 35 biomarkers, I'm only interested in these 3. Can you select these 3 and pivot them wide there for me to incorporate into my export? We're working on that. But yeah, this is one of the big things we're trying to accomplish here, integrating study and sample manager modules. But now with the new CDMS, it looks like this is going to be streamlined out of the box. And so we look forward to learning more about this and incorporating that into our work. I failed to mention, we just learned a couple weeks ago and got some of our initial instruction on using assays. It was good to see another group showing how they've been using assays. A lot of our data is historical, and we've uploaded things that we've already had at present, but moving forward, I think that we're going to build out an assay module there that integrates both with the sample manager and the study modules to surface analysis data in both of those locations to help us find and use the data that we need. Then the last thing we're working on is onboarding new studies within our division, and then helping other divisions learn how to use LabKey for their research. We got a bunch of divisions here at Cincinnati Children's. It's a big research institution that really interested in using this platform. And we've been very pleased so far with everything that we're seeing and the support that we've been receiving. I just want to thank everybody that's helped out with all this effort. Patrick Lonnie is literally amazing. He's done so much in terms of building within LabKey and just being familiar with SQL and XML to push data back and forth between different modules. Mindy and Dave from the IS team have been fabulous, and we're really happy with the support of Phil Hagedorn and Tracy Glauser and the CCRHRF leadership. Our project manager, Joe, has been helpful every week working with us and getting back in timely fashion when we have questions. And Hannah and many others have just been really helpful in helping us learn this whole platform. So if you have any questions or thoughts, suggestions, criticisms, we are so happy to learn. Please raise them now or shoot me an email. Be happy to learn from your experience or share what we've stumbled across.
Thank you, Dr. Standage. Great presentation. We do have a question. At what point do you anonymize patient data? Is that handled at the hospital level, outside of LabKey, or otherwise?
So yeah, so we have data are anonymized at each site. And this is one of the things that we will build out later. Our CRCs are clinical research coordinators are very separate from the lab team that manages the samples and the analyses. And so they actually maintain all of the keys apart from LabKey. And I'm learning that we could probably build some of that structure within LabKey itself and give them a portal to maintain, because right now they literally live as password secured Excel files on a secured shared folder, out on a CCHMC server. Maybe not the best solution, but each site maintains the keys for their anonymization algorithms.